Just have no pressure. You know, this was this thing is not working. Thank you. It's a sad day for her. She's getting everything set up. She's gonna do some test first, but this is the data order that recorded it. Oh. I felt like I was really so bring a long sleeve shirt. I'm so glad I had it. More.
No.
Yeah, I shouldn't have brought it.
So
Because I wore a long sleeve shirt. That's immediately. Yeah, I carried one around yesterday too.
Oh, yeah.
I don't know why I brought it.
I never get out. I'm above that.
Yeah, you're an employee.
I'm like, wait, can I get it back in the office?
That's what I was thinking. You got it.
She think about respect. She knows how to treat people with that.
And it's like in the way, so when you open the door, it's like
Literally, like,
walk
it's
around
on, it's like on wheels, yeah, like moving. Yeah, you could have moved forward. But you're just not very safe.
and maybe that's what I was thinking.
Thank you. Hold up.
Better? No. We went on Tuesday night, we didn't get my sticker.
Oh, really?
Yes, sir.
I saw that, that's why I wanted to make it. No, we're gonna talk about that. Is the cure for cancer? No. No, we're gonna talk about this.
Maybe. I was stressed out.
No, that'd be pretty good. Yeah. The booth was awesome. And those earlier, they might be actually.
I can't walk in.
Should try to.
Started contacting her.
That's his daughter. Many stopping her.
I know they're probably gonna try to pay it
Yeah.
for sure. And
She was too keen on sound. Well, she kind of was. She was just trying to stop herself from falling out of the hill, which was easily preventable.
Yeah.
She was like, Mama, help. I'm like, you can just like stop falling. You can stop sliding.
And you still have another.
Yeah.
Yeah.
So.
So the question is,
Okay.
so that's what's best for you.
It does because
That's the only
if
one.
I, you went through this, then we case and I'm gonna.
So where do I?
I don't want some foot.
Jennifer,
It's not gonna fall.
but then Holly also. So, hey, Jennifer's back for the rest of the week.
Yeah.
Taking care of our technician.
Yeah. No, we're not having the symptoms of that.
Yeah, he's he's actually doing some kind of diet.
And so you can.
Yeah.
See where you can just get the here's.
Is there any way that I can use the remote? Is there-
Yes.
I didn't have class.
I will, I can do it tomorrow, because that's, yeah. I just want, I don't like that.
I didn't try that, but it tasted like a shit.
Already.
I wonder if because of the size of this file, if I can better to put it on right up in the very.
Yeah.
There was some warning about an unlicensed product. That's actually, yeah. They don't. I have a whole bunch of embedded videos, and that's just writing presentations like itself. Kind of neat.
The first
So
one.
that's where all the videos.
was gonna try to do that. But once we got that, he doesn't need to, but started catching up on the side, which probably usually
Well, they also.
Yeah, it could just be undefined.
I had fun last year.
So she's highly rated. Yeah, they had it.
Yes. I don't think Okay, thank you all for coming today. We have someone from Clemson with us, Dr. Childress, who is an associate professor and the undergraduate program coordinator in the biology department. He got his BS in biology from the University of Tampa, an MA in zoology from the University of California at Berkeley, and a PhD in biology at Florida State University. He's an evolutionary behavioral ecologist studying the impact of climate change and behavioral adaptations in marine animals. His teachings and research focus on understanding the impact of habitat loss on conservation of marine communities. He enjoys training graduate students, undergraduate students, and teachers in the methods of experimental marine ecology. Thanks for joining us.
Thank you. Very great to be here. I appreciate you guys coming to attend this seminar. What I want to do today is take you on a field trip to the Keys. That would be my preferred way to teach you what I'm going to try and share with you today. And I try and do that for as many people as possible. But I want to start off by sort of setting the stage and talking a little bit about why tropical marine communities are endangered. If you look at any of the reports that have come out recently from the IPCC or any of the supplementary reports, they identify shallow water marine communities as one of the most threatened communities on the planet. The predictions regarding the sustainability of coral reefs in the face of rising sea surface temperatures is bleak. And there's a lot of statistics thrown out there like coral reefs could disappear within the next 50 years. So this is, without hyperbole, one of the most important communities to study to understand the impacts of climate change. So just a little bit of background. This is a picture that was taken by the Ocean Agency. The Ocean Agency, Richard Beavers was a collaborator on the Chasing Coral films. He's the one who did the XL Catlin surveys, 360 surveys, where they swam around on reefs with giant 360 cameras all over the world so you could do virtual dives. And as he explains in the film Chasing Coral, when you see a picture like this, you go, so beautiful. It's such an incredibly and beautiful environment. It's diverse, it's got all these incredible species, etc. But if you're a knowledgeable coral ecologist, you see a lot more problems in this picture than you do solutions or beauty. For example, this should be entirely covered in the surface of this should be covered in polyps of this giant star coral, but it's not. A large portion of this structure is covered in calcareous algae, which are invading since the coral tissue has died. You never find soft corals growing out of the top of hard corals. That is a consequence of the hard corals disappearing and a shift in the community to these more soft coral species. This subtly change in color right here, that coral is screaming out that I am dying. I am losing my zooxanthellae because of the heat temperatures. So until you have sort of a familiarity with these systems, it's often very hard. And I have this true with all my undergraduates that come to the Keys and they're like, this is the most beautiful thing I've ever seen in my entire life. But that shifting baseline has made it harder and harder for people to understand what we're seeing and how it really has changed through time. And so if we go back as far as Connell's classic paper in 1978, coral reefs and tropical forests were sort of put forward as these incredibly diverse and stable communities upon which it would take a great deal of disturbance to sort of move it from its climax community, which was dominated by competitive exclusion. But if you had disturbances, diversity would initially increase. The more you had a storm or a frequency, it would knock out those dominant players in the community and more diversity would occur. And it's not until you got really, really far down the line of extreme disturbance would there be any negative consequences in terms of community structure. We now know and have modified these thinking in some important ways. Some of the work and sort of focus of the folks like Emmett Duffy and others who are thinking about this problem of biodiversity and stability, the relationship between biodiversity and stability. They've really focused on going the metabolic route by trying to understand how productivity is the key feature which maintains the biodiversity stability relationship. And one of the things that is definitely happening in our warming ocean is that we're changing the metabolism of all of these creatures. Everyone has an accelerated rate of metabolism, and so is that going to accelerate the rate of herbivores faster than the algae? Is that going to accelerate the rate of predators faster than the herbivores? What are those consequences to community structure? So this is a classic paper. It's Garner's 2003 paper sounding the alarm that coral reefs in the Caribbean are dying. Phil Dunstan down at College of Charleston has nice photo series of the same reefs of Carryfort Reef in the Florida Keys, showing that we went from this incredibly 60% coral cover dominated by acropora species to these broken up acropora piles to this rubble pile that practically has no live coral left in a lot of these places. And so if you just sort of extrapolate Garner's lines from where we were in 2000, I shouldn't even be talking about coral reefs today. They should have gone extinct. We should have hit the zero here, but we haven't. So what has happened since 2000 that has prevented us from the inevitable? And can we learn from that to project forward and prevent the catastrophic predictions that we're going to lose these communities by 2050? One thing we know for certain is we lost structure. The loss of acroporid corals, the loss of brain corals in both the Caribbean and in the Pacific, with the loss of the table species of corals, have resulted in massive increases in fleshy algae. One paper published in Science described this as the slippery slope to slime, that we are losing corals and shifting our communities to algal dominated communities. And there's been lots of papers about phase shifts and whether these are alternative stable states between a coral rich community and an algal rich community. But the interactions that go on in a coral reef are complex. They span size classes from the tiniest plankton to some of the largest invertebrates on the planet. That these fish that use the coral reef during different parts of their lifetime can even cross through 4 orders of body size within their own lifetime. So we've got this complex system of giant organisms and tiny organisms tracking the flow of energy, tracking the flow of matter in these systems is a monumental task. And it's one of the huge challenges in understanding the dynamics. If we knew what the interaction strengths of these relationships were, we could predict, well, let's pull hard corals out of this and see what happens. We don't know those interaction strengths well enough to make those kinds of predictions. But that doesn't keep us from trying. And so one of the goals is to try and understand, if we were to identify the major drivers of coral reef ecosystems, can we understand what the climate change push is doing as a system, as a whole? Not just how is climate change affecting corals, but how is that corresponding effect of corals affecting the invertebrates, affecting the fish, affecting the whole structure of the community. And it'd be great if we were just dealing with climate change, right? But unfortunately, we're also dealing with intense fishing pressures, and we're dealing with decreasing water quality. So it's like, which way do you want to die? Do you want to die from nutrient pollution, overfishing, or from water being too hot? So there's been a massive effort to try and shift from measuring these individual interactions to trying to study this process as a whole. And I'm sort of on this same journey because I started out as a behavioral ecologist, and therefore I was very focused on understanding interaction strengths on pairs of species. And you'll see this as I go through my talk and talk about some of my prior projects. But the idea is now I'm to the point where I'm really beginning to think, how can I use all that information to study this from a broader perspective? So most of my work's been conducted in the Florida Keys. The southern tip of Florida is a chain of islands, which unfortunately are connected by a road. And that has meant that there's been far more human impacts in the Keys than there would have been had they not been connected by the road. But it's also undergone various management strategies for protection. So Everglades National Park was established in 1934, and it includes one of the largest estuaries in North America. But it's huge, incredibly productive estuaries known as Florida Bay. Dry Tortugas National Park has Fort Jefferson, a Civil War era fort that's out there. This is part of the Keys that is not connected by the road. And this is now protected for many species, including fishery species. There's John Pennycamp Coral Reef State Park. So the state of Florida recognized that they had a valuable resource as early as the 1960s and set aside protections off of Key Largo. Then Biscayne National Park was added to the portfolio. And then finally, in 1990, the Florida Keys National Marine Sanctuary was established and sort of linked all these other government jurisdictions. That's great news. A lot of this area is protected. It's bad news if you're a researcher because you need an FWC permit, a national park permit, and a Florida Keys National Marine Sanctuary permit to work in this. Okay, so I want to sort of bring you along some of the studies that I worked on in the Keys, starting with what I did my dissertation work on and my early work once I arrived at Clemson, which was lobster sponge coral mortality interaction. So Caribbean spiny lobsters are one of the most important marine species in the Florida Keys. They support a huge commercial fishery. We harvest about 5 million pounds of lobsters every year like clockwork. And no matter what has happened, climate change, loss of habitat, changes in fishing regulations, we still land 5 million pounds every year. Fortunately, we're in good shape because we're on the upstream end of the Gulf Stream, which brings us our larvae, and we can harvest every single reproductive adult with no consequence to our population. We are in a unique situation there. But I was interested in this transition that little lobsters go through. Here's an even smaller one here. And you can see when they're really small, they blend in with this fleshy algae. And they start out their life solitary. And they quickly move to associating with other juveniles, and they're often found crowded together. Literally, this lobster is standing on top of this other lobster in a shell. So here is telemetry to monitor and follow the behavior of lobsters. So this is a pinger that's literally attached with super glue to the back of the lobster. They will retain that until the next molt, but it gives us the ability to actually track the movement and behavioral associations of lobsters as they move around. So as I was saying, lobsters have this larval phase, which connects their populations throughout the Caribbean. The larval stage is about 9 months. So literally, there's no population structure, or almost no population structure throughout the Caribbean, because the larvae in the Keys could have come from Belize or Cuba or the Bahamas or the Lesser Antilles. They metamorphose and transition to the hard bottom as these tiny juveniles that live in algae. And then they undergo this really cool transition from being a solitary individual to being a gregarious individual about 1 year of age. And it's this year of age when they're incredibly vulnerable to predators. And so the initial thinking was this transition to sociality was to defend against predators. You were safer in a group than you were by themselves. And so we were interested in, well, what happens if sponges and corals are impacted by climate change? How might this influence Caribbean spiny lobsters and the protection that they hopefully are getting from these? So we established sites along the Middle Keys, all the way up from the southern tip of Florida Bay, all the way down off of Marathon. And we've tracked spiny lobsters, how they were using habitats, et cetera, how gregarious they were. And literally within the first month of my dissertation work, the beautiful crystal clear waters of Florida Bay became pea soup. And this was the first of a series of phytoplankton blooms that were going to devastate this estuary. This Synococcus bloom lasted for about 4 months. And when the waters cleared, all of the sponges that the juvenile lobsters were using were dead. This algal bloom actually clogs the sponges and they suffocate. And so we thought, well, if there was ever going to be a way we could demonstrate the importance of structures of these little guys, we should go out and measure this impact because it's got to be huge. Those little vulnerable lobsters need that structure in order to survive. Fast forward till I'm a professor here at Clemson and I have a graduate student studying lobsters in the Keys, and sure enough, another algal bloom occurs, 2007. So this has been a repeated phenomena of these cycles of Synococcus blooms. The sponges will grow back for a few years, another bloom will hit, it'll kill all the sponges. So this time, at least, we were better prepared. We had been studying the dynamics of lobsters in these areas before and after the impact, and the bloom only affected half of the sites, so we had by chance the most perfect before after control impact design that you could ever want, but could never actually design if you tried in life. This graph simply shows that sponge abundances before in blue were pretty similar across all the sites, and sponge abundances in red were pretty much truncated wherever the blooms occurred. So there should be a corresponding change in juvenile lobster abundance. So we went out, we measured these, and sure enough, if you just count the number of structures of any type, sponge, coral, solution hole, seagrass edge, that lobsters were using, there were, the number of structures had decreased by 1/3 after the impact. But the number of lobsters actually increased. So as much as we thought we understood how this system worked, right? Big die-off. The number of lobsters on our sites were higher than before the sponges died. And one of the things we learned is that lobsters have an innate way of dealing with heterogeneity in their habitats, and that is they use conspecific cues to find shelter. So if the sponges are all gone, they can still use their nose to figure out, you found a shelter, I'm going where he's at, and they can relocate to an area that still has shelter. So because these areas that we were measuring still had shelter, they were actually refuge for those lobsters that were displaced by the sponge dive. So much for even a simple link between a resource and the abundance of organisms. So what did we learn? What we learned was eutrophication, which is driving those algal blooms, is having a huge effect on the competition, causing negative effects on things like sponges, and that sponges provide protection, but lobsters have this behavioral compensation which is allowing them to get to sort of overcome that kind of impact. I'm happy to report we're still landing lobsters at the levels we were before, even though you can measurably say that a third of their nursery habitat's been disturbed. Okay, second vignette is parrotfish and macroalgae. This is sort of the classic one. When I started, back in the Keys around 2010, I said, I've only got 10 years of 15 years of field work left. What do I want to spend my time on? And I thought, well, if I can help save the coral reefs, that's a pretty worthwhile thing to put my efforts in. At the time, really, it wasn't until about 1998 that the state had really been paying attention to the status of coral reefs. They had done some surveys in the 70s and 80s, and you can see the percent cover of coral was way up in the 40%, with a large portion being these purple acroporid corals. But by the time I got to Clemson and started working in the Keys, 4 of those coral species were on the endangered species list. So 2 acroporids, as well as the pillar coral and the giant star coral, were already on the endangered species list. But you can see that same leveling off that I showed you before, right? It got down to about 10% coral cover and it's just leveled off. And these graphs only go out to 2010. So what has happened in the decades since? And that's where we've been concentrating our efforts. Some reefs look like this. It looks like literally every ounce of the bottom is covered in this Dictyota algae. And this is algae that's sometimes, 20 centimeters deep on the bottom, and literally filling in every crack and crevice. But then on other reefs, when you go to it, it looks like this. And the big difference is this big, beautiful super male parrotfish right here in the middle. This is a queen parrotfish. But you can see that the surface of the substrate is now maybe got a light filamentous turf algae on it, but it's not that thick Dictyota algae. So what's the dynamic between the herbivores, the algae, and then these remaining weedy coral species, which is what we're working with left? Mustard gill coral, Porites, and Ciderastria are the 2 major reef-building corals that are sort of left surviving. A number of people have speculated on what the ecological pathway is that links parrotfish or herbivores to corals to sustainability. And this idea that there's alternative stable states. And here's a summary by Peter Mumby and Bob Stanek that basically suggests if you start with a negative feedback, or let's start here with increased macroalgal cover. You increase macroalgal cover, it causes coral recruitment to decline. There's no place for the larval corals to hit bottom and settle. That reduces coral cover, which in turn affects structural complexity, which fish use to survive predation. So then you have too few parrotfish recruiting to the system. You have reduced grazing intensity, which leads to further increased macroalgal cover. And the alternative is, if you can reduce macroalgal cover in some way, you will recruit more corals, there'll be increased cover, the increased cover will increase structure, structure will make more fish will make more grazers, and so you go one of 2 ways. If you're right here in the middle, you either go toward having no coral and all algae, or if you can tip it in the right direction, you go for having lots of coral and very little algae. So it's an unstable community equilibrium that should be tipped one way or the other. And so the idea is, as practitioners of conservation, can we move the needle? Can we get it across this threshold into the self-perpetuating feedback, which will save the ecosystem? At least it's a goal, right? And so again, using sort of a 3-group diagram, we have algae competing with corals. We have corals influencing the herbivore fish populations. Some of these can be negative effects, like the reef fish feeding on the algae, or even the feeding on the corals. Some of these might be positive effects through energy consumption or production. Some might be protection, preventing the next trophic level from eating. Okay, so there are all these direct and indirect interactions to be assessed. So we established 16 pairs of reefs in the Middle Florida Keys that go from inshore reefs that have really high coral cover, like 25% coral cover, and also tend to have lower visibility, higher turbidity, because they're closer to Florida Bay. They're nutrient rich, they have lower visibility, but they have really high coral cover. And as you go further and further offshore, you have less and less corals, the reefs get flat, but they get perfectly clear. So now you've shifted from a high hard coral environment to a high soft coral environment. And you've got different dynamics with regards to temperature, salinity, and turbidity. So these are the guys we chose to study. This was my first field project on fish. And of course, I chose an easy group to identify and to recognize because it's not bad enough that we have 10 different species of parrotfish, but they have to have 3 different appearances in their lifetime, from the juvenile to the initial adult to the super male phase. So it's like learning 30 different species of fish to try and study this group. But they're amazing and fun animals. This is a scene that you will see nowhere else in the Caribbean. This is a mixed school of midnights and rainbow parrotfish on one of the reefs in the Keys. And because these fish are so big, they're really hunted heavily in most other places in the Caribbean. They're protected in the Florida Keys. But you can see, if I can catch up to the school, they were moving pretty fast. When they stop to feed, man, there are mouths scraping the rocks everywhere, and they just move in mass through these environments. And you can see how much of the surface is completely cleared of algae by the efforts of these parrotfish moving through the environment. And sorry, I don't have sound for this one, but you can hear them crunching. It sounded like they're eating breakfast cereal. Okay, so this is a phylogeny from Choate that shows that our parrotfish in the Caribbean, 6 of them are Scarus species, 4 are Sterosomas. One interesting evolutionary fact about these guys is most of the sister taxa for these guys are in the Pacific, rather than these guys having a shared common ancestor that was in the Caribbean. And almost all of these taxa, their sister taxa is across the Isthmus of Panama. But we took these 10 species and we wanted to see if their diets aligned with their phylogenetic relationships. So what these diagrams show is the proportion of bites. I made Kylie follow more than 2,000 parrotfish to do this. So we followed more than 2,000 parrotfish and she recorded every bite they took. And then we separated it into the proportion of bites in these 6 different categories represented by the 6 different colors. This bottom bar represents the frequency of those food categories across the area that we are working. So it's mostly turf with a small amount of fleshy algae, small amount of calcareous algae and maybe 15% of that fleshy algae. Then there's hard coral, soft coral, or sponge, hard coral, soft coral as the composition. So algae is about 60% of the bottom, but it's about 80 to 90% of the diet. So parrotfish are herbivores. They will take bites on corals, but they're not out there looking for corals. In fact, their rates of taking bites on corals is below what you would expect if they were randomly taking bites of the substrate. But there are 3 functional feeding groups. One is this group of the Sperosoma that have preferences for all 3 types of algae. They have them pretty equally in their diet, fleshy, turf, and calcareous. Another functional group of feeders are those big ones that you just saw in the video that are focusing primarily on turf and calcareous algae, but they don't eat the fleshy algae. So unfortunately, that big, massive biomass of blues and rainbows and midnights, they're not the ones clearing out that Dictyota algae. So it's important to understand what they're feeding on before you start making predictions about how they're going to influence the algal communities. This middle group includes 3 species of Scarus and the one most abundant species of Sparosoma, and they primarily are turf-only feeders. And so now that we have sort of a better idea of the functional groups, rather than grouping all the parrotfish together to see what their effects are on algal communities, we can group them by functional group and see if the functional groups are having specific targeted effects on their algae. You just throw it all together, you do all the parrotfish and all the percent covers on all the sites that were censused all the years. There is absolutely no relationship between parrotfish abundance and algal abundance. So this again was not what we were expecting to find. This again sort of belays the difficulty upon which you can isolate one pairwise interaction in a complex system and expect to be able to see anything from this. There are lots of reasons why this relationship falls apart. One is there are different kinds of algae in different locations. So when you lump them all together, you're just mixing up reefs that are high algae and low algae, and they are not replicates of one. Another consequence of this is they change their diet. They aren't like, I gotta have turf, I only eat turf. They're not vegans. They are, they're not restricted to one diet. They will change their diets when they're in different locations, and there's different opportunities. They change their diets from the juvenile phase to the adult phase, and from the adult phase to the superadult phase. So even within the lifetime of a parrotfish, when they're all swimming around as one school of juveniles, all of the juveniles across all 3 functional groups have the same diet. By the time they get to the adults, that's when we see the functional differences in their diets. So we turned our attention to the other half of the equation, which is, okay, so if we can't measure the effects of parrotfish on algae, can we measure the effects of algae on coral? So here we are doing our very first transplant of corals in 2012. We used a plumber's epoxy to attach to fragments of coral that we received from the marine sanctuary in our permit to place corals out in controlled areas. And just like any good manipulative experimental ecologist, our first thing to do was to put a cage around it so that we could prevent anybody from eating our corals, right? So here we are naively thinking, okay, now we've got this incredible design, we're gonna put a cage around it, we're gonna prevent the parrotfish from eating the algae, we're gonna really get at and test this ecological interaction. So what did we find? Well, what we found was it didn't matter for these corals if they were in the cage or out of the cage. Some of them had algae, some of them didn't have algae. It was completely unrelated whether the cage was open or cage was closed. It also was very apparent that algae came and went with seasons. Algae came and went on some sites earlier and other sites later. There was no consistency in the relationship between the algae and the corals, with one exception. We did this over the time period from 2012 through 2015. And 2014 and 15 was one of the most severe marine heat waves that hit the entire planet. It's the one that pretty much caused 25% mortality in the Great Barrier Reef. We also saw above average temperatures indicated from this satellite data that NOAA collects. And these colored hills here represent the metric that is now the standard for measuring coral stress. It's called degree heating weeks. And it basically, anytime the temperature is above the 90th percentile of the range of temperatures, what happens is you're accumulating heat stress. And that's multiplied by time, so you get accumulating degree heating weeks. If you go 4 weeks, so if you're in the red or the orange here, that's you're about to bleach. Once you go beyond 4 weeks, you're likely to bleach. And once you go into the darkest red, you have a really strong probability of bleaching after 8 weeks. So we were able to actually track those corals through this. So we had individually marked corals. We could see that before the bleaching, they were fine. They went through this heat stress. They lost all their zooxanthellae, but they didn't die. They got their zooxanthellae back. They recovered. The second time they went through the bleaching stress, many of them showed higher resilience. So the ability to actually say, okay, this amount of heat's going to kill the corals doesn't work if you survive the first one. The corals often uptake different zooxanthellae that have higher heat stress ability, heat stress resilience, and as a result are primed to survive higher heat the next time around. Thank goodness corals are a community of organisms. They may not be able to change plastically as fast, but their symbionts do. And that's really important. And actually, some further work on coral bleaching has discovered that corals do upregulate different heat stress genes in the presence of extreme heat that suggests there is both a plasticity within the coral and plasticity within the symbionts. So the good news is that bleaching does develop a resiliency. You notice that some of these corals, there's more algae than other. The negative consequence of algae that we were found is that corals that had contact with fleshy algae were more likely to bleach. Corals that were not in contact with fleshy algae were less likely to bleach. So the effect of algae on the corals was not to kill the corals, but to stress them. And that stress showed up when you added another stress, which was the warming water. So what we found is that, again, if you overgrow algae, you do put a stress on the corals. The corals also are experiencing the stress directly from the environment. And what's going on with the parrotfish seems almost irrelevant, or at least we haven't revealed what the connection is. We don't know whether this is actually resulting in more parrotfish, less parrotfish. It was still kind of a open question. So, Kylie graduates, Kara comes in as the next graduate student, and she's interested in how the structure of the communities influence reef fish abundance and diversity. So not to try and make anyone feel bad about their project, but Kara decided that she would study all the different reef species and the consequences of structure on everything in the community. She quickly realized that that was probably not going to be a successful master's thesis. But I at least give her a lot of credit for the desire to want to do something big. I know Kyle's smiling because he went through this whole process with Kara, getting her to sort of focus in on something that you could really do a thesis out of. And that focus was really going to emerge quite by chance. Kara ended up studying a project she didn't set out to study, because a lot of times in ecology, you go where the data leads. And so while we thought we were going to be studying the entire reef fish community within this structure, and we set up our sites and did the same thing, we had, of course, now we had 5 years of data on the abundance of corals and algae on these sites that gave us a good starting place for her project. But she was interested in structure, so we were motivated by these 2 pictures that I took 2 decades apart. This was a picture that I took as I was finishing up my PhD in the Keys. And you can see these grunts happily hanging out underneath a beautiful elkhorn coral, branching 3 foot tall, creating this great shady environment that grunts so love. And 20 years later, they're doing the same thing. They're just hanging out under a soft coral. Same grunts, same relationship in the environment. They just substituted one structure for the other. So that led us to ask the question, does the structure shape matter? And of course, as a good experimental ecologist, we had to create artificial structures that we were then going to put out and test and see whether reef fish responded differently. How Kara managed to stand on her head while she was doing this one remains to be seen. Anyhow, while we were setting this up, we kept noticing, he's like, what's wrong with these corals? They don't look well. And it was something that we were like, I don't know what's going on. We've never seen this before. No one had reported on this. We were scratching our head. So we decided, okay, there's something going on. Maybe we should follow as controls, healthy structure, disease structure, structure that had already died, as well as soft corals in the natural environment. And it's a good thing we did. So we oftentimes would set up GoPro cameras on these structures. We would allow them to take pictures in the absence of a nosy diver, and then we would come back and we would take the recordings from that to estimate fish abundance. But what we discovered when we weren't in the water looking was this: that some species were actually coming to these diseased corals to feed. And in particular, this four-eyed butterfly fish really seemed to like the diseased corals. And this was not something that we had predicted or expected in any sort of way, but immediately gave us the out to direct Kara, like, okay, you have a new thesis. You're going to study the influence of coral disease on butterfly fish abundance, okay? And sure enough, when we went back and we looked at the photos, their precision upon which they were actually feeding on the line of tissue that was being stuffed from the corals was quite remarkable. These guys are targeting wherever the corals are peeling off that layer of tissue. And if you think about it, if you're a butterfly fish, it's hard to get soft tissue out of a coral. Your mouth is structured from millennia of evolution to be able to do so, but you still have to suck pretty hard to get tissue out of the coral. It's a whole lot better if the coral's just sloughing tissue and you can just go along and slurp it up. So we found, one of the tragedies of this disease outbreak, which we didn't know what it was, and so they called it stony coral tissue loss disease, and that became, by abbreviation, Skittle-D, and everybody's been trying to overcome that abbreviation. It's like Skittle-D sounds like a cartoon character, not a plague which is destroying 22 species of corals throughout the Caribbean, but it is a plague. And unlike a lot of the other coral diseases that have occurred, this affects far more species and far more families of corals than any of the previous diseases, and we still don't have a causative agent. It started in Biscayne Bay in 2014. It moved about 40 miles a year down through the Keys. It reached our sites in 2017, and it's reached the Dry Tortugas now as of last year. It has also spread to the Virgin Islands. It's spread to Belize. It's spread to Cuba. It's spread all over the Caribbean. It affects the major mound-building corals that were left. Remember that 10%? These were it. Montastria, Solanastria, the brain coral, Copophilia, Corbicella. Those were the 10% we had in the Keys, and this disease was killing them. So we started paying attention to what was happening with all 4 species of butterfly fish on these sites. Butterfly fish abundance went from less than 1 per 500 meters square to like 9 per 500 meters square during the year that the disease was active. After the disease passed in 1 year, it was back down to where it was before. So there was this rapid abundance increase in the butterfly fish during the time that disease was moving through. I'm sorry, the rapid increase is disease corals, and is only about a 2.5-fold increase in butterfly fish. But there was correspondence between disease and butterfly fish abundance. What Kara was able to show is that during the year when there was active infection, the butterfly fish had shifted their focus from feeding indiscriminately in the environment to targeting disease corals. So they were preferentially feeding on disease corals when that tissue was available. The following year, we went back, exact same corals. Unlike other places in the Keys, our disease corals survived. 19 of the 20 disease corals survived, but the butterfly fish were now not targeting the ones who had been infected previously. They went back to sort of a broad feeding across all the different corals. And so this momentary observation that butterfly fish were shifting their diet quickly to take advantage of this opportunity led us to ask 1 of 2 questions. Butterfly fish, are they good for corals? Or are they bad for corals? Because one thing that wasn't clear, and that is, it's possible that butterfly fish were actually removing the pathogen as it was spreading across the surface of these corals. And it may be that butterfly fish were slowing the rate of progression of disease, or they were taking disease particles and pooping them out over a whole bunch of healthy corals and spreading disease. So that was the sort of next phase. And what Kara did is she did a laboratory study which She just got accepted for publication that basically showed that when parrotfish or butterfly fish are present, disease transmission is slowed down. It's not completely cured, but it doesn't move as fast across the surface of the coral if those butterfly fish are picking along that eating front. So again, there's this influence of climate change affecting this in terms of stressing the corals, and then it might have something to do with nutrients. We don't know because we don't know what the pathogen is yet. And we have this consumption effect that we thought was negative, but now maybe it's actually positive. So once again, a simple study, no clear answers. So what have we learned other than marine science or marine ecology is hard? What we've learned is that maybe trying to study one relationship at a time is not the best way to go. Maybe trying to understand the pattern happening throughout the community simultaneously is a stronger approach. So our study started with one of those Synococcus blooms in 2012, followed by the heat wave of 2014 and 15, followed by the disease outbreak of 2017 and 18, followed by another heat wave in 2019 and 20, and then just for fun, we added Hurricane Irma right in the middle of this. So on Connell's diagram, I think we're way at the right-hand side of the diagram in terms of disease or disturbance frequency, right? So diversity should be going down, resiliency should be going down. How can the corals be hanging on when this is the onslaught of disturbance, which is going? So if we overlay the degree heating weeks measures from the satellite, you can sort of see where those heat waves are coming from the rise in degree heating weeks during those years. If you add to it the salinity graph, there's a relationship with salinity as well. Years that have high heat are calm winds, lots of evaporation. So salinity goes up and heat goes up because it's calm, and turbidity goes way down. So when you measure turbidity, it has the opposite cycle. Turbidity is going up when it's windy and it's churning the water up, it's releasing heat, and a lot of fresh water is coming into the system. So we have these abiotic drivers that are asynchronous in their effect on the community. If we add on to that now, what's been happening in the percent cover of our algal species, we begin to see some patterns. During the heat wave, turf algae increased. And it was so high during the second heat wave, maybe not so much. Fleshy algae also showed a rise during the heat wave and another rise during the heat wave as well. So maybe there is a link between the heat production and those algae. And it might not be the heat, it might be the clear, calm water, which is allowing the algae to do really well during these years. What's happening to the soft corals and the corals? Soft corals were on the rise until Hurricane Irma. And Hurricane Irma's main effect was just shear those soft corals off right at the bottom. So there's this huge drop in soft corals after Hurricane Irma, and they've been working their way back. Corals, hard corals, showed a little dip in that first heat wave, but those that survived showed no change through the second heat wave. They had developed some degree of resilience. 10% hard coral to begin with, 1 decade later, still 10% hard coral. So again, resiliency when we weren't expecting resiliency. What about the fish communities? So our three most abundant and representative of our different functional groups, functional group 1, stoplights, peaked during the heat waves. Red bands, which are functional group 3, peaked during the heat waves. And rainbows, it's a little hard to tell because they're so much less abundant. But the heat waves, whether it's the clear water, whether it's the lack of nutrients, whether it is the warm temperatures, for whatever reason, we're seeing the abundances of the parrotfish during those times. So how do you do this? Well, you put it all together into some sort of a path analysis. We're trying to pull out from all of these simultaneous data what's going on. And so degree heating weeks are positively related to the parrotfish abundances and the turf algae. We tried to flip this around and put the algae up in the second level, and the parrotfish model was having none of that. Okay, it is not driven by heat through the fish to the algae. It's driven by heat through the algae. If there are any fish effects, they're secondary to the effects on the algae. When you look at fleshy algae, it's actually a positive relationship with the red band, but that's likely just a spurious correlation rather than any measurable effect. But there is this huge positive relationship between stoplights and corals. And mostly it's because stoplights like to live inshore where all the corals are at, and they don't like to live offshore where there aren't corals. I don't know what to make of that, whether that's a driver or just simply a correlation that is really, really strong. When you look at the covariances between these, there's a positive covariance between the parrotfish species, which I might have thought was going to be a negative covariance. But the reason why it's not is that spatially there's a negative covariance between these, but temporally there's a positive covariance between these. In terms of hard coral and fleshy algae, yes, there's definitely negative covariance. Turf algae and hard coral, definitely negative covariances. But hard corals are still persistent. And finally, how about heat directly on corals? Weakly, non-significant negative relationship, suggesting that the major way in which heat affects the corals is through the production of turf algae and then having the negative consequences affecting the corals. So by using these sort of approaches, we might be getting a little bit better than our species by species interaction to explain what's going on. So now I've replaced all the other factors with heat waves on all sides that are affecting this. Okay. Our future goals are to develop a model to predict coral transplant success. So after Kylie graduated with her PhD, she started a nonprofit in the Keys that now takes certified scuba divers out to actually assist in coral transplanting. So she's become a coral practitioner using what she's learned from these to make a difference. We are also interested in the relationships between structure and fish diversity. We're going to complete Kara's thesis long after she's graduated. We are going to keep working on that to answer that question. And also, we're trying to develop recommendations for best restoration strategies in the Keys. There's a huge effort to save 7 Florida Keys reef. It's called the Mission Iconic Reefs program, and they're throwing all their efforts into saving these 7 reefs by putting all of their concentrated efforts in one place. I also have a new graduate student, Kia Payton, who is adding fishing impacts right to the center of our diagram in the form of lobster traps, which get lost at a rate of 100,000 traps a year are lost by the fishery. And so those traps go on to kill fish or become substrate for algae or potentially influence communities. But these traps are so abundant that they're near every one of our sites. And we want to know what the impact of those are. And if you do marine debris removal, what are you removing with them? And what are the consequences of that? We also have a marine science outreach program that Randy Sims runs that is called Something Very Fishy. And the goal of this is to increase ocean literacy in elementary school kids and to empower them to have an interest in STEM careers to help save the oceans. And we have been successful. We recently did a virtual dive with our VR headsets, and the kids got to go to a dive on the reefs. But these kids want to save the planet, and we're not doing them a service to tell them that they can't do that or that there's no hope. And so it's really, really important that the message is, yes, you can save the planet. And we use the corals as a good example. For all intents and purposes, the corals should be gone by now, but they're not willing to give up. And so neither should these kids. And that's sort of the message we want to get. With that, I want to thank a ton of people, Kylie and Kara in this diagram, he and Randy that are watching by Zoom, as well as all the funding agencies for our work through the years. And I'll be happy to answer questions if I can figure out how to turn off the, that I'll just leave it there until I do that. But I'll be happy to answer questions. Yes.
Given your experience, for those of us coming into this field, what do you see as the things we can do that will impart the most positive impact going forward for the future generations? And like, where can we make that biggest impact in your?
I would say the way to make the biggest impact is include as many people in what you're doing as possible, in whatever way possible. Whether that is communicating your research on a web blog or a public forum, giving talks at non-traditional, non-academic places like the Garden Club or your child's elementary school classroom. Anything you can do to talk to people about your passion for conservation, your passion for science, you're making a positive impact. Then when you get in an academic position, invite those undergraduates to help. Recruit graduate students that are like-minded, that they're gonna want to invite graduates or undergraduates to help. The Creative Inquiry Program, I can't say enough about it. This work couldn't have been done without them. So that's a huge opportunity while you're here at Clemson.
Michael, I was curious about going way back to the start of your talk, lobster habitat interactions you saw with those soft sponges disappearing and lobsters becoming really aggregated and remaining habitat. How does that interact with the fishery that exists within? Are fishers able to more accurately target them, or are they not aware that's going on? Is there any sort of interactions there?
There is a huge interaction, but it has nothing to do with the sponges. It has to do with the way the fishermen fish. So in the Florida fishery, they're allowed to use juveniles as bait. And so every trap they put out is a den like, come to me, this is a shelter, I'm already here. And that's actually how they exploit the adults, is by loading the traps up with juveniles that then attract in the adults. The consequence of this is that we've measured a decline through the years on how willing lobsters are to be attracted to lobster food. We don't know whether that is a fishery-induced phenotypic change, or the other possibility is that lobster odor is more attractive if you're well-fed or just eaten. And so if you're a scrawny, starving lobster, you're not very attractive. And we've tried to convince the fishermen this matters to their traps because they put a short in it, they're starving. They leave the short in there 2 weeks, already the catch is going down. They leave the short in there 4 weeks, and it's now a repellent in terms of keeping adults from entering the trap. So in terms of like targeting areas where there's a lot of structure, the traps far outweigh the abundance of the natural structure in this environment. So a lobster is moving across Florida Bay is going to encounter sequentially, a coral, a trap, a trap, a trap, a trap, a sponge, a trap, a trap, a trap, a trap, a coral. So there are far more traps than there are natural structures in many places.
Yes, I just cannot take out the eyes from your shirt. How many species are on the shirt?
Yeah. Who'd like to calculate the beta diversity of my shirt? I should add that if I add my mask to it, then we increase the diversity further.
So Michael, I'm curious, from a technology standpoint, clearly what you were doing back in the distribution tools you had and now it changed. I'm just curious, are there data that you're collecting now that you wouldn't have been able to because of the technology, and how you see that changing in the next 10 years? What's on the forefront that you guys are looking at going like, this is going to change what we can do?
Yeah, the satellite data is phenomenal, and the data booted data. So that degree heating weeks and all of that salinity and turbidity profile data I gave you was from the NOAA Chica Rocks data booth. And that having access to that, because I mean, yes, we took our YSI out and we measured the salinity and the temperature on the day we sampled. But that's probably not enough to know why the algae is like blooming in 2014. But if we have an entire year's integrated heat profile for that site, now we have the kind of data that's really going to be meaningful. So I think like automated data from satellites and buoys is huge. Second of all, there are big pushes now to standardize all these sampling methods, so that whatever reef you're on, however many fish you're counting, however you're doing an estimation of coral cover, it's done in the same way so that we can all combine our data sets and have a much better profile across the broader spatial scale. And then advanced processing, right? The ability to do more complicated statistical modeling that deals with covariance matrices and things like that. Those are all, I think, big advantages now in trying to tease these things.
Are there pretty good comparison studies going on throughout the Caribbean, or are there big, or also are there spatial gaps like some areas of the Caribbean, like the Lessers, maybe where, you know, those countries just can't do the work?
Yeah, there's always gaps. There's always gaps, and there's been some effort to do sort of assessments and identify reefs that are critical to preserve throughout the Caribbean. And we've used a lot of particle tracking oceanographic models to identify places where would be good sources of larval recruits to be released from in order to reach the broader number of places around the Caribbean. So there are some efforts underway to sort of prioritize where conservation efforts should be. And of course, oftentimes the most pristine areas are the ones that we haven't fully exploited yet. And we're going into some developing community and saying, you know what? No more fishing here. We're going to set aside your reefs for the preservation of the reefs that the rest of us have all overfished. And so then there's the social challenges in terms of using that data and working with those partners. And really, it's convincing people you can make more money from people coming to watch the fish than you can from fishing fish. And once you can get people to start thinking that way, like these fish are money because people are going to come and pay and see it. And if we leave them and protect them there, they're going to keep coming and paying to see them. Then we might tip the balance in favor of meeting the economic demands and the preservation needs. But a lot of big data crunching and remote data sensing are the technologies I see as moving forward. Yeah.
Could you talk a little bit how you started something squishy and maybe how much time you spend on it?
Way too much. It was by chance. I'll put it all on Kylie. Kylie was giving a talk about her coral parrotfish work at the Ollie Center. You guys familiar with the Ollie Center? It's the Osher Lifelong Learning Center, and it's mostly retired folks or people who just want to learn things and have a lot of free time. Well, Kylie got invited to talk about her dissertation work there, and she decided to pair it with a showing of Chasing Coral. So she showed the film and she talked about her research and she talked about the plight of coral reefs. And the first question one of the participants said was, what are you doing to get into schools? And it was just, you could just see the look on her face. It was just heartbroken because we've been talking about it. We've been putting it in as broader impacts every time we put a grant in, but we hadn't yet figured out how to do it. And so that was her answer. We want to, we don't know how to do it. And a woman walked up to us after the presentation and said, I know how to do it. I'm a songwriter, playwright from Australia. I grew up on the beach. We used to have this traveling puppet show for kids in Australia about reef health. Why don't we combine forces? I'll do the musical theater production, you guys do the science. And something very fishy was born. And it was so awesome because Kathy is so creative. Her songs are all, she has one called 2 Degrees. And it's all about, it doesn't sound like much, but 2 degrees of warming has all these effects. And she's won Children's Songwriter of the Year in Australia multiple times for her songs. And so she came and joined us and came to our CI meetings. And she's like, I know nothing about marine ecology or corals except what I've read on the web. And so it became this partnership where we taught her marine ecology and she taught us how to present or to make it engaging or to get students involved. And that's how it started. So I'd like to tell you it was brilliant. I had this plan and I just, just came out of it. I created it all myself, but it was happenstance and chance. And again, it goes back to this talking to people outside of academia. And had we not done that, had she not agreed to do that talk, we might have never started something very efficient. So I think that's the take-home message I would say. And then once you do a project like that, people were like, I'm in. So people from Science Ed said, I want to be a part. People from the Performing Arts Department here at Clemson were our co-PIs on the latest NSF grant. If all goes well, we'll be putting the production on in the Brooks Center. We're moving forward with this. I got my first South Carolina Arts Commission grant, which is not something you normally would see on a CV, right? But there's a ripe field for connecting kids with an understanding of climate, because the concern is teachers don't feel comfortable talking about this scary issue. And you need some way to address that in a way that is within the comfort zone of the teachers. And everybody's behind saving the ocean. And so just by reframing that just a little bit, we're going to save the ocean. Who wants to help? Everybody's hand goes up, right? It's not political. We decoupled it from the politics of climate change. But if you save the ocean, you're going to have to do so by addressing climate change. So that's the strategy we've been using. I think it could be employed successfully in any conservation goal. You want to do this conservation project, you're going to inadvertently make the planet better. Thank you so much. I appreciate it.
I believe we do have the
Can I just go ahead and end?
Let's go.